Hi everyone, this is the first video in the Unit 4 Microbiology. In this particular video, we are going to talk about microscopy. So before moving into the technical details of the microscope, we are going to talk about two important scientists who made wonderful contributions in the field of microscopy. The first person is Robert Hooke. Robert Hooke lived in the period of 1635 to 1703. During 1660s, he made and used a compound microscope, which was like one of the finest microscope at that particular time. By looking into different species starting from insects, then cells, he published a book called as Micrographia, which was one of the top selling books at that time. He made and illustrated minute details of the things that he was observed under his microscope. And Robert Hooke also coined the term cells, which he observed from the organisms that he was looking under his compound microscope. The second scientist we need to know is Anton von Leeuwenhoek. So Anton von Leeuwenhoek was the person who first observed a living microorganism under the microscope, and he used a magnification microscope and discovered the unicellular independently living microorganisms, which he called as animalculous, because at that time the microorganism term was not coined yet, so he just referred it as animalculous. All his findings he shared with the Royal Society of London. And Anton von Leeuwenhoek was the person who first made a handheld microscope. That is, he just grinded the lenses and he prepared his own handheld microscope. So now we know these two people, how their contributions made help in the field of microscopy. So now we will see what is a microscope and why we need this particular microscope. So normally with our naked eyes we cannot see the objects that is less than about 100 micrometer. So if you want to see an object that's less than 100 micrometer, you need a help of some external tool. So that tool we call it as microscope. And this microscope is nothing but an optical instrument consisting of a lens or a combination of lens for making an enlarged or a magnified images of minute objects. So that's the basic definition of a microscope. And the microscopes can be broadly classified into two categories. The first one is light microscope and the second one is electron microscope. These two classifications are based on the types of source we are using in these microscopes. We'll talk more about these two types later. But before moving into these two types, we want to know about two important characteristics that all the microscopes should have. The first one is magnification. So magnification basically means it's an ability to enlarge the images. The second character is resolving power. So resolving power is the ability to show the detail of the image that we are observing. So we'll see in detail about this magnification and resolving power. So as I already mentioned, magnification is like increasing the size of the object that we are observing. And with the help of this magnification, we can enlarge the image size with greater size. But the problem is this magnification beyond the resolving power is of no value, because the larger image will be less distinct in detail and fuzzy in appearance. So we can't see the objects very clearly. So that's where comes our next character, that is resolving power. So resolving power is defined as the power of the objective able to separate distinctly two adjacent points. So it is the function of the wavelength of light used and the numerical aperture of the lens system which contributes to this resolving power. So if we have a greater numerical aperture, greater the resolving power of the objective and finer the detail it can reveal. So basically this magnification and resolving power both has to relate with each other so that we get a very best quality of the image. Now we are going to see about the parts of a microscope. So there are different parts available in microscope, and it's going to be different from one microscope to another microscope. But broadly we can classify these parts into three categories, that is magnifying parts, mechanical parts and illuminating parts. So what is a magnifying parts we can see. So magnifying parts basically are the objective and the eyepiece lens. So if you can see the image here, so this is the simple structure of a microscope. So you can see here there are like two eyepiece lens, or we call it as ocular lens, through which we are going to observe the image. So this eyepiece lens in the top and the second objective lens, that's you can see here there are like different ranges of these objective lens based on the level of magnification it can create. So these two lenses we call it as the magnifying parts. The second one is mechanical parts. So mechanical parts actually does not create any magnification, or it's not going to provide any light source, but these mechanical parts are forming the basic structure of your microscope. For example, we have different mechanical parts like the base, the pillars, the curved arm of your microscope, then the body type, the coarse adjustment, the fine adjustment, the mechanical stage, and the revolving nose piece. You can see here, so this is your revolving nose piece. So this revolving nose piece is going to help you to adjust the different objective lens that you want to choose for your particular sample. Then this is your mechanical stage. So this mechanical stage is going to provide you the space or the stage to hold your specimen or sample. Then you have here the two adjustments. So one is, we call it as coarse focus. Okay, that is the larger knob. So using this coarse focus, you can adjust in a very greater detail. The second one is fine focus. That is the smaller knob. So if you want to adjust like very minutely, you can use this fine focus. So these fine focus and coarse focus are basically like the adjustment knobs. Okay, like zoom in, zoom out. You want to adjust the details level. Okay, so like how we use in a camera. Next, you have a XY stage knob. Okay, so you can see here in the bottom. So you have a stage knob. So this stage knob is going to help the stage move in the vertical direction. Okay, if you want to lift it up or lift it down, you can use this XY mechanical stage knobs. The last one you have here in the bottom is a rheostat. So rheostat is to adjust the light intensity. Okay, because you are going to pass a light source to observe your image. So if you want to adjust the light intensity, you can. So now these parts we call it as the mechanical parts because they help in the magnifying parts and illuminating parts. The last third category of the parts in the microscope is illuminating parts. So illuminating parts consist of three things: the condenser, the diaphragm, and the illuminator. So you can see here the bottom one, that is the illuminator. So illuminator is actually responsible for illuminating the light source. So based on the type of source used in the microscope. So in the case of light microscope, it's going to be light, and in the terms of electron microscope, it's going to be passing electron beams. So apart from illuminator, you have the diaphragm and the condenser, which is also going to help in passing the light towards the sample specimen that's available in the stage. So these three major parts, that is magnifying parts, mechanical parts, and illuminating parts, form the different structure and parts of a microscope. In the next video, we'll discuss on the types of microscope, that is light microscope and electron microscope, and its subdivisions. Thank you.